Welcome back everyone. Today we will study a very simple and interesting topic, which is fluidity of plasma membrane. How the fluidity of plasma membrane gets affected by its own properties and by some external factors. We will start this lecture by a very simple concept of science, alright? That what happens when you boil something, when you provide heat to a solid. What happens? What is the effect of heat on any substance? If you provide heat to a solid will start melting. And what is melting? Melting is nothing but increase in distance between the molecules. Alright, so solid will change into liquid, and if we provide more heat, this liquid will change in its vapors, in gaseous forms. So what is the change brought about by heat? Heat increased the kinetic energy of the molecules. This increase in kinetic energy increases the molecular motion between these molecules, and these molecules will start migrating away from each other. The distance will increase, and we say the solid melts into liquid, and the liquid boils and evaporates into its vapors. Alright, so this concept applies in cell biology as well. So what happens to fluidity of lipid bilayer? What are the factors that affect the fluidity of lipid bilayer? There are mainly three factors, and assuming I am here talking about plasma membrane, but only the lipid portion of plasma membrane. Alright, I am excluding the protein portion for the simplicity of this video. So for this particular video, lipid bilayer and plasma membrane is thought to be synonymous. Alright, let's move on. So there are mainly three factors that affect the fluidity of lipid bilayer. The first factor is very obvious, temperature. We will talk into details how this temperature affects the fluidity of lipid bilayer. Alright, but let's talk about other factors, or the names of other factors as well before that. Nature of lipid also affects the fluidity, whether we are using a phospholipid or we are using sphingolipid. Do we have cholesterol in between these lipids? Do we not have cholesterol? All these factors affect the fluidity. The chain length of fatty acids will also impact the fluidity of membrane. If we have shorter chain length, but will be the fluidity, and how this fluidity will be affected if chain length increases. Unsaturation means double bond. How many double bonds we have? That also impacts the fluidity of lipid bilayer. There is a term in case of temperature dynamics that we use, the melting temperature, Tm, or transition temperature. It is that temperature at which 50% of membrane is in solid state and 50% of membrane is in gel state. Means 50% of membrane is compact and 50% of membrane is in molten state. Alright. We will not be using this term as such, but this is the term which we will be defining using a simpler language in this video. Alright, let's move on to temperature. What is the effect of temperature on the fluidity of membrane? Alright, what will happen if I increase the temperature? In this first figure right here, we have our normal membrane, and in second figure is what happens when I increase the temperature. Okay, we increase the temperature. As we said in the beginning, by increasing the temperature, we increase the kinetic energy between the molecules. We must also know about what stabilizes the membrane. What are the forces that stabilizes the membrane in this state? These forces are non-covalent in nature, and mainly the force which is involved is van der Waals forces of attraction. These van der Waals forces of attraction are present between these lipid heads as well as these fatty acyl chains. These are the attractive forces. Van der Waals forces of interaction. If we increase the temperature, molecular motion will increase and these attractive forces will be decreasing. There will be less attractive forces so that these molecules will be free to move around. This is the main concept. Okay, molecular motion is increasing, the distance will be more, and van der Waals forces of attraction will be less. So membrane will be more fluid. The fluidity increases with increase in temperature. Alright. To move on to other factors, other properties of membrane, we will assume that we are keeping the temperature constant to better understand the concepts further. At constant temperature, before moving on to other factors, if we increase the distance, there will be more space in between two phospholipids. Okay, so permeability of membrane for gases, for smaller molecules, for water will also increase. Let's move on to some other factors. At constant temperature, what will be the effect of nature of lipid? What will happen if we have membrane made up of phospholipids, and what will be difference if we have membrane made up of sphingophospholipid, okay, or sphingolipids. So you must already know that phospholipid has two fatty acyl chains and sphingolipid has only one fatty acyl chain. Okay, What difference will it make? Where will be more forces of attraction? Okay, so see these two figures and you will find out that more forces of attraction will be in case of phospholipids due to two fatty acyl chains, and there will be less forces of attraction in case of sphingolipids because there will be more distance between these fatty acyl chains. All right, so in case of phospholipids we have more attractive forces. As a result, we have lesser molecular motion, we have lower distance between these monomers, and we have lower permeability. Whereas in case of sphingolipids, there is only one fatty acyl chain, and these fatty acyl chains are distant from each other, as a result of which van der Waals forces of attraction will be lesser. There will be greater molecular motion, greater distance, and higher permeability. Okay, again, for moving on to next factor, we are again assuming the temperature to be constant. At constant temperature, what will be the effect of the length of fatty acyl chain? If we have smaller fatty acyl chains like 16 carbon palmitic acid chains, and if we increase the chain length to 18 carbon or 20 carbon chains, but changes will be observed in case of smaller fatty acyl chain and in case of longer fatty acyl chain. In which case will be higher attractive forces? Let's see, we have attractive forces like this, and here in longer chain we have attractive forces like this. Longer chains have more carbons, have more chain length, have more surface area. That is why the attractive forces will be more in longer chains and attractive forces will be lesser in smaller chains. We are assuming the all other properties, the nature of these fatty acids, the nature of these polar heads are exactly the same, so that these other properties won't influence the fatty acyl chain length and membrane fluidity in this case. Okay, in case of smaller fatty acyl chain, we have lower van der Waals forces of attraction. As a result, we have higher molecular motion. As a result, we have higher fluidity. In contrast, we have more surface area, which results in higher van der Waals forces of attraction, which results in lesser molecular motion, more compactness, and lesser fluidity. Let's move on to next factor, which is degree of unsaturation. By unsaturation, we mean double bond. Triple bond is not present in biological tissues or biological cells. All right. What will happen if we have a lower or no unsaturation and we have higher unsaturation? The unsaturation or double bond brings about a kink in the structure. Okay, what is the significance of this kink? This kink increases the distance between these fatty acyl chain, and if greater the distance, we have decreased attractive forces. Whereas in case of a lower unsaturation, we have these fatty acyl chains in close proximity. That is why the attractive forces will be more. Okay, in case of lower unsaturation, we have more attractive forces, we have lesser molecular motion, as a result of which this membrane is not very fluid. We have lower fluidity. In contrast, we have lower attractive forces due to this kink present due to unsaturated double bonds. We have more molecular motion due to this kink because distance between fatty acyl chains increases. We have more fluidity and we have more permeability as well. All right. What is the effect of configuration of this double bond? Okay, this double bond can exist in two configurations, either in cis configuration or in trans configuration. Let's talk about cis configuration first. The cis configuration brings about asymmetry. The symmetrical structure will not be formed. This asymmetry will disrupt the structure. All right, and as a result of which van der Waals forces of attraction will be less because the structure is irregular. All right, if the structure is irregular, distance is more, molecular motion will be more, and if molecular motion is more, we will have more fluidity. In contrast, trans structure brings about symmetry. If the structure is symmetrical, we have higher van der Waals forces of attraction. Due to higher interactive forces, we have lesser molecular motion, we have more compactness, we have lesser fluidity. So that's the effect of double bond, the state of double bond. So the cis double bond increases the fluidity and trans double bond increases the decreases the fluidity. All right. What is the effect of cholesterol? So cholesterol is a type of planar molecule which incorporates or which is present in between these phospholipids. All right, it is a planar molecule. It is very large molecule having multiple these ring-like structures. All right, aromatic structures. And cholesterol is a rigid molecule. Cholesterol provides rigidity to plasma membrane, and if it provides rigidity, it decreases the molecular motion. It increases the van der Waals forces of attraction. How will it increase the van der Waals forces of attraction? It will provide more surface area for fatty acyl chains and polar heads to interact, and it will form a more compact structure. Molecular motion will be lesser and fluidity will also be lesser. Okay, that is the effect of cholesterol on the fluidity of the membrane or lipid bilayer. Okay, let's talk about the practical applications of this concept. Okay, the main practical application is resistance, or we say adaptation. How do animals adapt themselves? Climate is not same everywhere around the world. All right, there may be some snowy regions where temperature is very low. There are desert habitats where temperature is very high. So how do the plants and animals adapt themselves at cellular levels? That is the application of this concept. Okay, let's talk about chilling resistant plants, the plants that grow normally at lower temperature. So normally at lower temperature, van der Waals forces of attraction will be higher because there is low temperature. So there will be low kinetic energy between these molecules. So we have higher van der Waals forces of attraction, which will lead to lower fluidity. But temperature may decrease as well. If the temperature decreases to an extent that membrane freezes, and there will be such a hindrance in the transport of molecules that cell may die. So how do these cells survive? How do these cells adapt to such situations? Let's see. What will these cells do for the survival? These cells will increase the membrane fluidity. And how will they increase the membrane fluidity? They will have to shorten their chain length. By shortening their chain length, the interactive forces or van der Waals forces of attraction will be decreased. If van der Waals forces of attraction are decreased, distance will be increased, molecular motion will be increased. All right. And second thing these plants can do is increase unsaturation, introduce more double bonds, introduce kinks. These kinks will increase the distance between these fatty acyl chains. Due to increase in distance, the attractive forces will decrease. Again, distance will increase and fluidity will be more. That's how the chilling resistant plants adapt. They shorten their chain length and increase their degree of unsaturation. All right. In contrast, what happens in heat tolerant plants or the plants which grow at higher temperature? Okay. At normal, what happens at higher temperatures is we have lower van der Waals forces of attraction because heat provides more kinetic energy to molecules. Okay. The fluidity will also increase because there is lesser attraction. There will be more kinetic energy and there will be more fluidity. So if the temperature further increases, the membrane may melt away and all the content may leak out. So how to avoid this situation? How do these plants survive? How do these plants adapt? These plants will have to decrease the membrane fluidity. And what is the way? What is the possible way to increase this membrane fluidity? How do these plants adapt? They increase the chain length. Okay. If we have more chain length, we have more attractive forces. And by more attractive forces, we overcome these situations. All right. And second thing we can do is we can decrease unsaturation. We need to remove the double bonds. We need to make straight fatty acyl chains so that distance between these fatty acyl chain is less and interactive forces will be more and fluidity will be less to counter the heat situation. So we increase the chain length and we decrease the unsaturation. That's how heat tolerant plants or plants which grow at higher temperatures adapt themselves. This is the practical application of this concept. The same kind of phenomena are seen in animals as well. And in case of marine animals, the fishes which live at warmer zones of sea and the fishes, the fish which lives at colder climates of the sea, these will have different structures in their plasma membranes to adapt to such situations. This is the practical applicability. Okay. That's all for today. We will be uploading more such lectures accordingly. Thank you.